For decades, most people pictured asteroids as dead things, cold rubble drifting through empty space, rock with no chemistry, no history, nothing worth studying beyond size and shape. But when Japan reached out and touched one, brought a piece of it home, sealed and untouched by Earth's air, the samples kept getting stranger. On December 5th, 2020, a small capsule dropped from Japan's Hayabusa 2 spacecraft and landed in the Australian outback, carrying material collected directly from the asteroid Ryugu 2 years earlier. Scientists opened the capsule expecting rock. What they found kept rewriting the story. The building blocks of RNA, more than 20 amino acids, a mineral that, according to everything we know about asteroid formation, should not have existed on this rock at all. And then, something alive, something no one expected, and for a brief, electric moment, something that looked like it might be the biggest discovery in human history. This is everything scientists have now confirmed was hidden inside the samples from Ryugu, piece by piece, discovery by discovery, and each one forced researchers to rewrite what a supposedly dead space rock could actually contain. Because by the time the analysis was finished, Ryugu was no longer just a rock. It was a time capsule, a chemistry set, and, unintentionally, a warning about how difficult it might be to ever truly recognize life that isn't from Earth. To understand why these discoveries mattered, you have to understand what made this mission different in the first place. Ryugu is a carbon-rich, primitive asteroid, a C-type object believed to be leftover construction material from the earliest days of the solar system. Roughly 4.5 billion years old, largely unchanged since formation. Most missions that bring rocks home focus on simply surviving the trip. Hayabusa 2 focused on something harder, keeping the sample pristine, never letting Earth's chemistry touch it before scientists could study it in a controlled environment. The mission launched in 2014 and reached Ryugu in 2018. Over the following months, the spacecraft carried out 2 separate touchdowns, briefly contacting the surface to collect material each time. But the more important sample came from the second touchdown. Engineers fired a small projectile into the asteroid to expose material from beneath the surface, material that had never been touched by sunlight or cosmic radiation, material that had spent billions of years shielded from the kind of weathering that alters rock exposed on the surface. In other words, not just a sample of Ryugu, a genuinely fresh look at the early solar system. On December 5th, 2020, Hayabusa 2 released a sealed capsule during a scheduled flyby of Earth, and it parachuted down into the Australian outback exactly as planned. From there, JAXA scientists handled the material inside state-of-the-art clean rooms, using sterilized tools to catalog individual particles one by one. The samples were sealed inside nitrogen-filled containers to prevent oxidation and contamination, then shipped out to research teams around the world under the same strict conditions. This obsessive level of care was not excessive caution. It was the entire point of the mission. Because if scientists wanted to know what a truly untouched piece of the early solar system contained, they needed a sample that had never been exposed to Earth's air, Earth's moisture, or Earth's biology. Then the labs opened the containers, and the story of a supposedly dead rock started falling apart almost immediately. The first shock came almost immediately. Ryugu was not chemically dead. It was packed with organic material, carbon-based molecules scattered throughout the sample in far greater variety than many scientists expected from a supposedly primitive space rock. Amino acids were among the first things researchers found. Initial analysis turned up around 10 different types, but as testing continued, that number kept climbing. Eventually, researchers confirmed more than 20 different amino acids inside the Ryugu material, including glycine and alanine, two of the fundamental building blocks used to construct proteins, the molecules that do almost all of the actual work inside living cells. Now, amino acids alone are not proof of anything extraterrestrial. Earth is covered in them. Contamination is always the first suspect, but this is where the story gets more convincing. Alongside the common amino acids, researchers also found rarer ones, isovaline and norvaline, molecules that are unusual on Earth but common in certain types of meteorites. And critically, these amino acids appeared in nearly equal left-handed and right-handed versions. Life on Earth almost exclusively uses one specific handedness. A near 50-50 split is a chemical fingerprint, a signature that strongly suggests these molecules were not introduced by terrestrial biology at all. They came from space, but amino acids were just one part of a much larger inventory. Scientists also identified aliphatic amines, hydroxy acids, dicarboxylic acids, and polycyclic aromatic hydrocarbons, compounds researchers described as similar to the complex carbon chemistry found in terrestrial petroleum. Nitrogen-bearing compounds showed up as well, adding even more complexity to what was supposed to be a simple, primitive rock. Here is why that matters. These molecules are not just interesting on their own. They are prebiotic chemistry, the class of molecules that exists one step before life, the raw material that, under the right conditions, can begin assembling into something more complex. Their presence points to something specific happening on Ryugu's parent body billions of years ago. Liquid water, likely warm, moving through rock, triggering chemical reactions that slowly built these organic molecules over enormous stretches of time. Suddenly, the picture of Ryugu changes completely. This was never just a lump of ancient rubble floating through the solar system. It was, in a very real sense, a chemistry lab. One that had been quietly running its experiments for 4.5 billion years, undisturbed, unedited, and completely sealed off from Earth, until a spacecraft reached out and finally opened the lid. Organic molecules were one thing. But then, researchers went looking for something far more specific, something far more meaningful. Not just chemistry that resembles life. The actual molecular letters used to write life's genetic code. Nucleobases are nitrogen-containing molecules that form the alphabet of DNA and RNA. There are 5 of them. Adenine and guanine, known as purines. Cytosine, thymine, and uracil, known as pyrimidines. Combined with sugars and phosphates, these 5 molecules become nucleotides, the actual building blocks that spell out genetic information inside every living organism on Earth. Without them, there is no code to copy, no instructions to pass down, no life as we understand it. In 2023, an international research team reported finding one of these nucleobases inside the Ryugu samples, uracil. Then, in a more recent study, a team of Japanese scientists went further and confirmed something extraordinary. All 5 nucleobases were present inside the Ryugu material, appearing in roughly similar amounts. And this was not an isolated result. The same 5 molecules had already turned up in the Murchison meteorite, which fell in Australia in 1969, and the Orgueil meteorite, which fell in France back in 1864. Then, in 2025, NASA's Pristine Samples from the asteroid Bennu delivered the exact same result. All 5 nucleobases confirmed again. But here is the detail that makes this even more interesting. These asteroids are not identical copies of each other. Murchison is enriched in purines. Bennu and Auger lean more heavily toward pyrimidines. Scientists believe this balance may be shaped by the presence of ammonia, a molecule that can influence which nucleobases are more likely to form. Each rock, in other words, is running a slightly different recipe, built from the same basic ingredients. Zoom out, and the implication becomes enormous. The genetic alphabet, the very components required to build DNA and RNA, may have been forming across the solar system billions of years before life ever appeared on Earth. Asteroids like Ryugu could have rained these molecules down onto the early planet, seeding it with the raw ingredients required for biology to eventually take hold. But not everything scientists found inside the Ryugu samples fit neatly into this story. Because one of the ingredients hiding inside the rock simply should not have been there at all. While studying the mineral structure of the Ryugu samples, a research team at Hiroshima University noticed something that did not belong. Using a technique called field emission transmission electron microscopy, they spotted a mineral called djicerite, a potassium-bearing iron-nickel sulfide embedded inside the rock. At first glance, that might sound like a minor technical detail. It is not. Djicerite is normally found inside enstatite chondrites, a rare category of meteorite that forms under extremely high temperatures in the inner solar system, close to the Sun. Ryugu formed nowhere near there. Its parent body coalesced from icy mixtures of carbon dioxide and water out at the cold, distant edges of the early solar system. These two environments should never overlap. And yet, here was a mineral from one showing up inside a rock from the other. One of the study's co-authors summed up just how strange this was. Its occurrence, he said, is like finding a tropical seed in Arctic ice. The chemistry only deepens the mystery. Laboratory experiments show that djicerite typically forms when potassium-rich fluids interact with iron-nickel sulfides at temperatures above 662 degrees Fahrenheit. Ryugu's history, as far as scientists understood it, never should have produced conditions anywhere close to that. So researchers were left with two competing explanations. Either materials from wildly different regions of the early solar system somehow mixed together during its earliest, most chaotic years, or Ryugu itself experienced small, localized pockets of extreme heat and unusual chemistry that no one had previously recognized. Early evidence leans toward the second explanation, but researchers admit they cannot be fully certain yet, not without further isotopic study. Either way, the discovery forces a rewrite. As the researchers put it, this single finding challenges the notion that Ryugu is compositionally uniform. This was never a smooth, evenly mixed rock. It was a patchwork, a chemically inconsistent object shaped by forces scientists are only beginning to reconstruct, and clear evidence that the early solar system was messier, more chaotic, and far more interconnected than the tidy textbook models ever suggested. Every discovery up to this point had rewritten some piece of Ryugu's ancient history, but the next one was different. This one was not about the asteroid's past at all. It was about something happening right now, inside the sample, on Earth. In November 2022, planetary scientist Matthew Genge was peering through a scanning electron microscope at a millimeter-sized speck of Ryugu when he noticed something odd. A minuscule, rod-shaped tendril of organic matter, unlike anything catalogued before, clinging to the surface of the grain. I thought, well, that's strange, Genge recalls. Then he found another, and another. For a brief window, he and his colleagues considered the possibility that they had just stumbled onto the most significant discovery in human history. Alien life, delivered to Earth inside a few grams of material retrieved from a space rock 300 million kilometers away. But the more they studied the rods, the more familiar they started to look. If these really were extraterrestrial organisms, they were behaving suspiciously like hungry Earth microbes. Over the course of a single week, the population grew from roughly 11 individual rods to around 147, nearly a tenfold increase, gorging themselves on the organic material inside the sample. Then, almost as quickly as they multiplied, they began dying off, collapsing under repeated bombardment from the same electron microscope being used to study them. A textbook boom and bust cycle. Explosive growth followed by rapid collapse. Watching that pattern unfold, Jeng and his team reached an uncomfortable conclusion. These were almost certainly invaders from Earth, microbes that had somehow colonized an extraterrestrial sample despite every precaution taken to prevent exactly that. Follow-up imaging using nanoscale X-ray tomography confirmed it. No microbes existed in the grain back in October 2022, when the sample was first examined. The contamination happened afterward, and almost certainly not inside JAXA's ultra-controlled facilities. It likely happened during a routine preparation step at a laboratory in London's Natural History Museum, where the grain was briefly removed from containment to be polished for analysis. "We were quite careful with it," Jeng explains, "but no one was as careful as JAXA has been, because we don't have the facilities. All it takes," he adds, "is a single bacterial cell landing on the sample. That is enough to contaminate it completely." The likely culprit, based on shape and behavior, is a microbe from the genus Bacillus, a hardy group of bacteria known for aggressively colonizing new and extreme environments almost anywhere on Earth. A definitive DNA identification was never possible, because the rods had already been sealed beneath a thin carbon coating applied earlier for imaging purposes. Jeng was candid about the outcome. "It's a bit embarrassing," he admitted, "but as a scientist, you have to tell the truth. It's important to report these things." The deeper significance goes far beyond one contaminated grain. It reveals just how relentlessly Earth life colonizes anything even remotely organic, even a fleck of asteroid handled inside supposedly pristine, sterile conditions. And it raises an unsettling possibility for the future. If our own instruments and spacecraft can seed life this efficiently by accident, we may eventually face samples from Mars or icy moons, where we cannot immediately tell whether we discovered alien life or simply exported our own. The Ryugu contamination was not just an isolated embarrassment. It is a warning for every sample return mission still to come. This problem is not new either. Decades ago, scientists concluded that bacteria linked to human nasal contamination had somehow survived aboard NASA's Surveyor 3 spacecraft, hardware that sat on the lunar surface for more than 2 years before parts of it were retrieved and brought back to Earth in the late 1960s. Technology has advanced enormously since then, but the underlying problem has never fully gone away. It has simply gotten higher stakes. Consider what is coming next. NASA's Mars Sample Return Program is designed to bring back material specifically chosen because it might contain biosignatures, actual evidence of past life. Mission scientists have stated they are preparing enhanced cleaning protocols and comprehensive sterilization procedures specifically because of this risk. NASA's Artemis 3 mission may return material from the icy, shadowed craters near the lunar South Pole, regions far more chemically active than the dry Apollo landing sites ever were. And JAXA is preparing to launch its Martian Moons exploration mission in 2026, aiming to return samples from Phobos, one of Mars's 2 small moons. The danger here is not abstract. If scientists mistake a contaminant for something that was actually part of the sample, the consequences ripple outward. As Genge put it, you start contaminating science with incorrect hypotheses. An entire research direction can end up built on a mistake nobody noticed in time. And here is the uncomfortable truth underneath all of it. Microbes are everywhere. No laboratory on Earth, no matter how advanced, can guarantee absolute sterility. As one geologist put it plainly, they are everywhere. From a scientific perspective, you just do the best you can. Earth life may be the most efficient colonizer in the known universe, and that makes contamination something that can be minimized, but never fully eliminated. So, could scientists someday be fooled, mistaking ordinary Earth microbes for something truly alien? According to one astrobiologist, probably not, unless alien life happens to look a lot like life from Earth. Put all of it together, and none of these discoveries stand alone. The amino acids, the genetic letters, the impossible mineral, the accidental microbes. Each one reshapes what Ryugu actually is, not a dead rock. A preserved fragment of the early solar system, carrying the raw chemistry of life. Evidence of ancient mixing nobody predicted, and, unintentionally, a live demonstration of how aggressively Earth life spreads the moment it gets the chance. And the same molecules keep showing up elsewhere. On Bennu, inside Murchison and Augay. The ingredients for life, scattered across the early solar system, possibly delivered to a young Earth from above. As humanity prepares to bring back pieces of Mars, the Moon, and Phobos, Ryugu already taught 2 lessons at once. The building blocks of life may be common across the cosmos, but recognizing genuine alien life, separating it from our own contamination, will be far harder than simply finding something that looks alive. The samples answered old questions, and left us with a stranger one. How will we ever really know what came from somewhere else?